Okay, so let us start today's session. Finish shared. So today we will cover carbohydrates part of the biomolecules. Biomolecules, all the foods belongs to this category. Even cell, tissue, organs, living organisms. General biomolecules that our food contains are carbohydrates, amino acids, proteins, nucleic acids, and vitamins. All of them we will cover. Even hormones we will cover in biomolecules. Today we will start with the carbohydrates part. So carbohydrates, they are produced by generally plants, and most of them are naturally occurring organic compounds. Examples are sugarcane, glucose is there, glucose, starch. So carbohydrate, as the name suggests, it is carbo means related to carbon, and hydrates means water. So carbon plus water, that leads to this name, that is carbohydrates, and generally simple ratio is 1 to 1, H2O, like C6H12O6, C12H20O11. Generally ratio is almost 1 to 1. Like in glucose, the formula is C6H12O6. In terms of water, we can write it like this, C6H2O6. So that's why it is a hydrate of carbon. That's why name is carbohydrate. Let us say sucrose. Sucrose is C12H22O11. So in terms of water, it can be written as C12H2O11. So hence these are called hydrates of carbon. General formula is CXH2O5, and here X equal to Y, or X may be not equal to Y. Most common sugar in home, that is sucrose, and lactose is also known as milk sugar. Now exception. So if we see acetic acid is not a carbohydrate, but yes, we can write it as hydrate of carbon, C2H2O twice. Even raffinose, that cannot be written as hydrate of carbon, but it is a carbohydrate. It indicates that all compounds fit into this formula may not be classified as carbohydrate. That means this definition is not a general definition for a carbohydrate. Compound that deviates from carbohydrates classification, that is acetic acid, glucose, sucrose, fructose, all of them, they are treated as hydrates of carbon. but acetic acid is also hydrate of carbonate, but it is not a carbohydrate. If we classify carbohydrates, carbohydrates are also called saccharides. Why they are known as saccharides? Because of the name sugar. They are sweet in taste. Most of them are sweet in taste, and those which are not sweet in taste, they are known as non-sugars. Classification of saccharides: they are monosaccharides, disaccharides. Oligosaccharides means they are varying from 2 to 10 units. Polysaccharides, carbohydrates that cannot be hydrolyzed, simplest carbohydrates cannot be further hydrolyzed. Give simpler unit of polyhydroxyaldehyde or ketone, they are called monoxide, because they are the one which are simplest unit of carbohydrate. That's why they are known as monosaccharides. There are almost 20 monosaccharides, leads to the formation of all the others, polysaccharides or disaccharides, glucosaccharides, glucose, fructose, ribose, they are all monosaccharides. Now, oligosaccharides, carbohydrates that yield 210 monosaccharide units on hydrolysis, they are called saccharides. They are further classified as di, tri, tetra, and so on. So, pentasaccharide, hexasaccharides. It depends on how many monosaccharides are there. Wide hydrolysis. Sucrose, maltose, even lactose, they are all disaccharides. Sucrose contains glucose and fructose. Maltose has 2 glucose units. Lactose has glucose and galactose. Polysaccharides, the one which yield large number of monosaccharide units on hydrolysis, they are called polysaccharides. Most common examples are starch, cellulose, glycogen. Now, monosaccharides, they can be classified on the basis of number of carbon atoms and functional groups. So, there are 2 types of functional groups, either aldehyde or ketone. Number of carbon atoms, their general term, aldehyde and ketone. So, if there are 3, at least 3 carbons should be there. 3 carbons, they are known as triose. If there is aldehyde group, then it is called aldotriose. If there is ketone group, that is called ketotriose. For 4, it is called tetrose, aldotetrose or ketotetrose. Same. Pentose, they are either aldopentose or ketopentose. Yes. Hexose, they are aldohexose and ketohexose. So, they are most common. Aldohexose is glucose and ketohexose, that is fructose. There is 7 heptose, aldo or keto. Ribose, is it monosaccharide, disaccharide, or glucosaccharide or polysaccharide? Obviously it is and monosaccharide. Polysaccharides, out of them, which one is polysaccharide? Now, classification of carbohydrates on the basis of functional group. You can classify it on the basis of taste and solubility, and nature of reaction. First, on the basis of functional group. Chemically, no, they are optically active. First and foremost thing is all carbohydrates are optically active. They rotate the plane polarized light either in the right or left. So if it rotates the light in the right direction, that is called dextrorotatory. If it rotates the light the left side, that is called levorotatory, and that can be predicted, measured by instrument that is called polarimeter. It is an experimental figure. You cannot say this one is dextrorotatory, other one is levorotatory. It is all experimental. So it is optically active, and there are more than one hydroxy group. They are optically active polyhydroxyaldehydes or ketones, or the compound which produce these units on hydrolysis. That is the definition, modern definition of carbohydrates, and it is universally accepted that it is optically active polyhydroxyaldehyde or polyhydroxytone, or the compound which give these units on hydrolysis. Chemically or functional group, so C6H12O6, we write open chain structure. So there is aldehyde group on the first carbon. Obviously, if there is aldehyde group, that should be on the extreme, means terminal carbon. This first carbon has aldehyde group, and sixth carbon, that is primary carbon. Primary alcohol is there on the, means we write these numbers in this way, that on the top there is most oxidized carbon, on the bottom there is least oxidized carbon. This is the order to write. First carbon has aldehyde group, sixth carbon has primary alcohol, and other carbon is there are different carbons, that is on the second carbon OH is on the right, third carbon OH is on the left, and fourth and fifth carbon OH is on the right. That you should remember, that which OH group is on the right side and on the left side. There is only one third carbon where OH is on the left. That you should keep in mind, and others are hydrogen. These are all secondary carbons or secondary alcohols. So there are 1, 2, 3, 4 secondary alcohols, one primary alcohol, and one aldehyde group. This is called open chain structure of glucose. So glucose has alcoholic group as well as aldehyde group. That's why it is called polyhydroxyaldehyde, PHA in short, polyhydroxyaldehyde, because there are more number of OH groups. That's why it is polyhydroxy, and only one aldehyde group, and that's why it is called polyhydroxyaldehyde. Now fructose, C6H12O6. So this one is aldohexose, because there is one CHO group, that is aldehyde group, and total 6 carbons. Fructose is ketohexose. There are again 6 carbons, but on the second carbon there is a keto group. That should find that second carbon, top second carbon, that has ketone group. Remaining carbon has alcohol group. First and last carbon has primary alcoholic group. And see again, third carbon, there is a difference on the third carbon only in both the cases. It is easy to remember that third carbon has OH group on the left. Rest are hydrogens. The secondary alcohols, 3 secondary alcohols, 2 primary alcohols, and 1 keto group. This way, fructose has again alcohols and ketone group. It is polyhydroxy ketone. Now let us see sucrose. Sucrose, when we hydrolyze it, we get glucose and fructose. One is polyhydroxyaldehyde, other is polyhydroxy ketone. So if we hydrolyze carbohydrate, we obtain polyhydroxyaldehyde and polyhydroxyketone. Carbohydrates are either polyhydroxyaldehyde or polyhydroxyketone, or the compounds like sucrose on hydrolysis give polyhydroxyaldehyde or polyhydroxyketone. So let us classify on the basis of taste and solubility. So on the basis of taste, they are either sugars or non-sugars. First, sugars. Sugars, first of all, they are crystalline, just like sucrose. They are sweet in taste and soluble in water. Now, non-sugar. So non-sugars are generally amorphous and they are tasteless, like starch, cellulose, and they are insoluble in water. So based on the nature of the reaction, you classify they are either reducing sugars or non-reducing. Those which have free aldehydic or ketonic group, they are reducing sugars, and if the aldehyde or ketonic group are attached, they are called non-sugars. Say in the aldehyde, aldohexose, say that CHO group is on the first carbon. So in aldo compounds, aldo carbohydrates, first carbon is called anomeric carbon, and in keto carbohydrates, second carbon, because CO group is at the second carbon, so that is called anomeric carbon. So in keto carbohydrates, second carbon that contains keto group is anomeric carbon, and in aldo carbohydrates, first carbon which contains CHO group is anomeric carbon. So reducing sugars, reduce failing solution and tolerance reagent. So all the monosaccharides, they are reducing sugars. Even disaccharides in which at least one group is free, either aldehydic or a ketonic group. If they are free, that is called reducing sugar. So obviously, if so, free means they are not joined by, because carbohydrates, monosaccharides, they are joined by glycosidic linkage. So if one of them is free, in aldose there should be first carbon that should be free, and in ketos second carbon should be free. If it is like that, either one of them, either first carbon of aldose or second carbon of ketose, if it is not joined by the other monosaccharide, that means it is okay. So maltose and lactose, say sucrose is not written here. Why? Because in sucrose, we will see it in the next slide, that C1 of aldohexose means glucose and C2 of fructose, they are joined together in sucrose. That's why there is no free aldehydic or ketonic group, and that's why it is a non-reducing. Sucrose is a non-reducing. Now non-reducing sugars, so they do not reduce saline solution or tolerance reagent. So those disaccharides in which aldehydic or ketonic groups are bonded, means they are joined together by glycosidic linkage, that is behaving as non-reducing sugar, and best example is caused by sucrose. Chemically, carbohydrates are polyhydroxy carbonyl groups. Carbonyl, aldehyde and ketones. Simplest ratio of carbon and hydrogen in carbohydrate is 1:2. Now, preparation and structure of glucose, that is in our syllabus. Glucose, what is the formula of the glucose? Is 6H12O6, and it is the most naturally abundant found in grapes, honey, even in blood. So now preparation. First in the lab and other is the commercial method. First lab method. So in the lab we can prepare it from the sucrose, because sucrose when it is hydrolyzed will gives glucose and fructose. Sucrose is C12H22O11. When it is hydrolyzed by water, the presence of acidic medium, get glucose and fructose. Now, how do we separate this sucrose, glucose from fructose? When we cool it and alcohol is added to the mixture, glucose being insoluble in alcohol rises out first, and fructose soluble in alcohol, that's why it remains in the solution. This way, as crystals of glucose are formed, they can be separated out by filtration. Now, commercially, it can be prepared from starch. Starch, it is a polysaccharide, C6H10O5N. When it is hydrolyzed, get glucose only. Excess of sulfuric acid is utilized by adding chalk powder. And sometimes there is a sugar which is brown or yellowish in color, and that colored impurity can remove by using activated charcoal. Activated charcoal is adsorbing those impurities. There is a phenomenon of adsorption. Adsorb colored impurities and separate glucose by cooling. Glucose, it is aldohexose and is also known as dextrose. Why it is known as dextrose? Because yes, it is dextro-rotatory. Can rotate the plane polarized light to the right, and it is monomer of many polysaccharides like starch, cellulose. It is most abundant organic compound probably. Can be written like this. This is the simplest one: CHO, CHOH4 and CH2OH. And in this CHOH, third carbon has OH on the left side. Other OH are on the right side. That you should remember. So first molecular formula was found to be H12O6, and if we heat it, the presence of HI is a strong reducing agent, so it can be converted into normal hexane. So it indicates that all the six carbons are in the straight chain. There is no branching at all. As it gives normal hexane, it suggests that all the six carbons are linked in a straight chain. So this is the first inference for its structure, that these six carbons are arranged in a straight line, that is indicated by the reduction of glucose by heating with hydrogen iodide. Second, glucose react with hydroxylamine and it gives oxime. Or even with hydrogen cyanide it can react to give cyanohydrine. That indicates it has a carbonyl group, because these are the typical reactions, characteristic reactions of carbonyl groups. NH2OH, no, CHO, it is like CHO when react with NH2OH, it gives CH double bond NOH. Wait for slide is visible to you? Yes, yes. Now, okay. Is equal to you. So now next is NH2OH. When NH2OH react with glucose, it gives CH double bond NOH, and rest part remains as such. So in this way we get oxime, goes oxime, that indicates there is a presence of CO group. Either aldehyde or ketone, both can give this reaction. Even if we have HCN, can also react with HCN to give CH, OH and CN. This two reactions indicate that this molecule has cyanohydrin, means it indicates that it has a carbonyl group. Gets oxidized to 6 carbon carboxylic acid, gluconic acid. When we treat it with mild oxidizing agent like bromine water, it indicates that carbonyl group is aldehydic. It has aldehyde group rather than other carbonyl group, means ketone. It has CO group on the first carbon, that is CHO, means aldehyde, that it indicates. See, we have, suppose when it is treated with bromine water, that is a mild oxidizing agent, we get gluconic acid. If we oxidize it with nitric acid, that is a strong oxidizing agent, it gives dicarboxylic acid, that is saccharic acid. So it indicates that is glucose as well as gluconic acid, both will give saccharic acid when we treat it with nitric acid. It indicates as one primary alcoholic group. Because it's commercially prepared from prepared by starch, and in lab it can be prepared from sucrose or cane sugar. Hydrolysis of sucrose produces glucose and fructose both. So the following is not a sugar. And if we do the acetylation of glucose with acetic anhydride, it gives glucose pentaacetate. It indicates it has 5 OH groups. Has 5 OH groups. It confirms the presence of 5 OH groups. How many acetic anhydrides are required? 5. We get each OH group is replaced by OCOCH3 group. Get glucose pentaacetate. All the 5 OH groups are attached to the different carbon atoms, because if 2 OH groups are attached on the same carbon, that is unstable. There should be 1 OH group each carbon. More than 1, they will lose water. Either we get carbonyl group or carboxylic. Pentaacetate, that indicates there are 5 OH groups on different carbon atoms. So exact spatial arrangement of different OH groups that we have already discussed, that which carbon has OH group on the left side. In glucose, carbon has OH group on the left side. Third carbon, that you should remember. See, configuration represent like gluconic acid and saccharic acid we can represent. First glucose, right straight line, write CHO at the top, CH2OH at the bottom, and then H on the second carbon, OH is on the right. The third carbon, OH on the left side, and rest are on the right on. That's it. This is the way to write. Just remember third carbon, that on the third carbon OH is on the left. Now gluconic acid, only first part, that is CHO, will change into COOH. Rest skeleton remains same. See, only first there is a difference on the first. In glucose this is CHO group, on gluconic acid there is COOH group. Rest part remains same. In saccharic acid, first and last carbon, that turns into COOH. Rest carbons remains as such. Now glucose, we can correctly name it as D plus glucose. Capital D plus glucose. Now what it means, what is capital D, what is plus? That plus is dextrorotatory, but what is D? D before the name of glucose represents the configuration. That means this plus indicates dextrorotatory nature. Plus can be represented as small d, but capital D doesn't mean it is dextrorotatory. It can be D negative sometimes. So capital D means on the second last carbon, OH should be on the right side. See, P and L have no relation with the optical activity of the compound, meaning we can have these notations. See, is relative configuration of a particular stereoisomer. Glyceraldehyde we take into account. See, in glyceraldehyde, it contains one asymmetric carbon. What do you mean by asymmetric carbon? That is chiral carbon. Chiral carbon means 4 different groups are attached to the carbon, and it exists in 2 enantiomeric forms. See, first and last, see, CHO and CH2OH, just like glucose, and on the second carbon there is OH on the right. So if OH is on the right side, it is called and if OH is on the left, it is not plus, it means capital D, and it means capital L. Okay, capital D means if on the second last carbon OH is on the right side, and if OH is on the left side, that is capital L. So most naturally occurring carbohydrates are capital D. So glucose is correctly named as capital D plus glucose. So capital D before the name represents its configuration, and plus represents dextrorotatory nature of the molecule. Part D plus glyceraldehyde. Now see this part of glucose. These two are same, CH2OH, HOH, CH2OH, HOH, and that's why it is called D plus glucose. Reaction of acetic anhydride with glucose indicates what it indicates. Yes, 5 OH groups of glucose and 1 CHO group, but acetic anhydride, it is option A. Reaction of acetic anhydride with glucose indicates 5 OH groups in glucose, and that should be on the different carbons. Correct representation of glucose is capital D plus glucose. And what is that representation for this part? The second last carbon as OH is on the right side, that's why it is called capital D. Now, cyclic structure of glucose. So, as of now, we have explained open chain structure of glucose. Structure 1, that is open chain structure of glucose, can explain most of its properties, but there are few properties which are not explained by open chain structure of glucose, and first one of them is, see, in all the reactions we have shown that it has free CHO group. That means it should give all the characteristic reaction of aldehyde. But 2,4-DNP test, this is the most common characteristic test for aldehydes. and that reaction is not shown by glucose. Even Schiff's test, that is also most characteristic test for aldehydes, again not shown by glucose. That means there is something wrong structure of glucose so far we have studied, because in those structures we have shown that CHO is free. Even it does not form this hydrogen sulfide addition product. It's also a characteristic test for aldehydes. That means aldehyde is group in the glucose is not totally free. That is not shown by open chain structure. Second, in pentacetate of glucose, again we have shown that CHO group is free, but it does not react even with hydroxylamine. Glucose react with hydroxylamine, but glucose pentacetate does not. That again indicate there is absence of free CHO group. Means CHO group, if it has, it is not totally free. And third is that glucose is found in two different crystalline forms. That is alpha D plus glucose and beta D plus glucose. That is also not shown by open chain structure. Alpha form of glucose, that is obtained by crystallization of concentrated solution of glucose at 303 Kelvin. This is alpha D plus glucose. See, rest part is same. Only there is a difference on the first carbon. On the first carbon we have shown so far that there is a CHO group which is free. But in cyclic structure we have seen that this carbon is bonded, form ring. So beta form, that is obtained by crystallization of hot and saturated aqueous solution at 371 Kelvin, and see again. Rest part is same. On the first carbon, OH, here OH is on the top. In the other case, OH was on the bottom. See, there is a difference on the first carbon only, and first carbon in aldoses is called anomeric carbon. There is a difference on the anomeric carbon, and that's why these two are called anomers. Alpha D plus glucose and beta D plus glucose, they are called anomers of glucose. So it was proposed that one OH groups may add to the CHO group and it forms cyclic hemiacetal structure. And this behavior that could not be explained by our open chain structure. See, this glucose where aldehyde is shown totally free, but in hemiacetal structures, CH on this carbon form hemiacetal. So if OH is the bottom is below, called alpha D plus glucose. If it is on top, above, that is called beta D plus glucose. These are alpha D plus glucose and beta D plus glucose. That I will show you how do we form it. This way, glucose forms 6 membered rings in which OH at the C5 carbon is involved in ring formation. Let us see. This one is the structure. See the first carbon and the fifth carbon. Fifth carbon gets attached to the first carbon. O here now, this O and this H attached to this oxygen, and that will be like this. So this OH on the first carbon, it is either on the right or on the left. So if it is on the right, that is called alpha D plus glucose. If it is on the left, that is called beta D plus glucose. One is alpha D plus glucose, other is beta D plus glucose. Okay. And these two structures exist in equilibrium, the open chain structure. And open chain structure is almost 0.02% only. Rest are alpha D plus glucose and beta D plus. To form its means Haworth structure formula, styrene is a cyclic organic compound. Oxygen, one oxygen atom and 5 carbon atoms. This is called styrene. Cyclic structure of glucose are represented by Haworth structures like this. So these are the Haworth structures. See, second carbon, look at the second carbon. HOH. So the one on the left side is written at the bottom. See, this is at the on the left side, it is written on the top. Again on the right, bottom. Fourth carbon. You know, fifth carbon is like this. See, 1, 2, 3, 4, 5, 6. The sixth carbon, it is CH2OH. First carbon and fifth carbon are attached like this. Second carbon, you know, it is HOH. On the third carbon is OHH. Fourth carbon HOH. So just look at the second, third and fourth carbon. Second, third and fourth configuration is same. On the other side there are H. Fifth carbon, CH2OH is always at the top. So it is the last carbon. It is written at the top. Now we left with first carbon. So on the first carbon, if it is HOH, tell me, this OH is on the bottom or below or above? Yes, in this case, I have written this. I mean to say this OH. What is the name of this compound? First tell me. Is alpha D plus glucose or beta? alpha D plus glucose. See, second carbon OH on the right, we write it on the right below. Third, the left, OH on the left, we write at the top. On the fourth, it is on the right side, is below. So if it is on the first carbon, if it is on the right, it is called alpha and it should be below. That means it is like this. Make corrections. It is like this, okay? HOH. So it is alpha D plus glucopyranose and OH on the is below the plane, you can say, on the first carbon. Now if it is instead of this, if we write OH like this, is OH on the left, so it is beta D plus glucose. Now this OH is on the top, this is beta D plus glucose, okay, and these two are called anomers. Yes, the two cyclic hemiacetal forms of glucose differs only the configuration of hydroxyl group at C1, and that is called anomeric carbon. Aldehyde carbon before cyclization, it is called anomeric carbon, and that's why it is called anomer. Two are anomers. OH on the anomeric carbon is below the plane, and that is called alpha D glucopyranose, and if it is on the top above the plane, that is called beta. These isomers, alpha form and beta form, that is 6 membered cyclic structures. They are called pyranose structure. Is alpha or beta? Alpha. Only we should look at this carbon, OH below the plane. That means the Fisher projection formula, OH is on the right. This is alpha D plus glucopyranose. Alpha D plus glucopyranose. This one is, yes, beta D plus glucopyranose. Right? Now epimers, these are anomers. So if there is a difference in configuration on any carbon except on anomeric carbon, any other carbon, if there is a difference in configuration, that is called epimer. These are diastereomers that contain more than one chiral center but differ from each other in the absolute configuration at only one chiral center. See, D-glucose, second carbon it is HOH, but in the D-mannose it is OHH. Means they differ at second carbon, not on the anomeric carbon. They are called epimers. They differ in configuration at carbon other than anomeric carbon. They are called epimers. Anomers differ at first carbon. Actually it is incomplete. Anomers differ at in aldoses. Aldoses they differ at first carbon, but in ketosis anomers differ at second carbon, because in ketosis CO group functional group is at a second carbon. Pyranoses pyranose 6-membered ring, 5 carbons, 1. Now structure of fructose. Glucose is aldohexose, fructose is a ketohexose. It is obtained along with glucose by the hydrolysis of fructose. This is sucrose. So tell me, what is this? Alpha or beta? Yes, it is alpha D plus glucose, or we can say alpha D plus glucopyranose. And you know what is the name of this compound? It is furan, right? 5-membered ring, 4 carbons and 1 oxygen. That is why this is, tell me, it is alpha or beta? Just look at OH. Yes, if OH is below the plane, that is called alpha. If OH on the top above the plane, that is called beta. So it is beta, again D, but now it is negative. It is laboratory. Fructose is laboratory. It is beta D negative fructoranose. Okay. Sucrose, the sucrose, see, this is called glycosidic linkage, COC, ethereal linkage. Obviously it is ether type linkage, but in carbohydrates this is called glycosidic linkage. Yes, we hydrolyze it, will break, and we get these two products. See, the glycosidic linkage is formed between C1 of glucose and C2, 1, 2, 3, 4, 5. C2 of fructose. So sucrose is non-reducing sugar or reducing sugar? Yes, sucrose is non-reducing sugar. Why? Because C1 of glucose and C2 of fructose both are involved in bonding. That's why it is called fructose. See its structure. The presence of plain polarized light. Is optically active, and see, this is same as D glyceraldehyde, D plus glyceraldehyde. So that's why configuration is D, but as it is laboratory, that's why it is as minus. and fructose. Is D negative fructose. See on the third carbon, in same case, OH on the left, CO. Here CO is anomeric carbon. See, in ketosis, second carbon is anomeric carbon. Is D negative fructose. Is also existing in cyclic forms. There is a linkage between C5 and C2. In aldoses, it was C5 and C1. Here in ketosis, it is C2 and C5. C2, 2, and this is C5. Again, there is glycosidic linkage. Alpha D negative fructopyranose. The structure was like this. Open chain structure was CH2OH. H2OH. The second carbon it was CO. The third carbon it was OHH. Carbon HOH. Fifth carbon again H. This fifth carbon and second carbon is the bonding oxygen. This hydrogen shift to oxygen comes OH on one side and CH2OH on the other side. See, CH2OH one side, OH on the other side. If this OH is on the right, it is called alpha. If this OH is on the left, that is called beta. See, third carbon it is OHH. Yes, OHH. Fourth carbon it is HOH. Yes, it is HOH. There is a difference, the second carbon. Got it? One is, yes, this one is yes, OH on the right, then alpha, OH on the left, beta. So if they form its furanose structure, see there is 5-membered ring, one oxygen atom. As I told that one which is on the left side, one which is on the left side is written one on the right side is written below the plane or above the plane. The right one is written below the plane. So that is, yes, is like this. On the third carbon it is OHH. Fourth carbon it is 1, 2, 3, 4. And fifth carbon, obviously last carbon is on the top like this. That is alpha D negative fructofuranose. Now tell me in this case. Yes, beta. And then it is OHH, HOH, rest are same. So it is beta D negative fructofuranose. These differ at 2 carbon. They are also ketose, they are anomers. Beta D negative fructofuranose. Furan is a 5-membered cyclic compound that you know. 1 carbon with 1 oxygen and 4 carbon atoms. Furan that we have discussed. So this is the Haworth structure, cyclic structure of two anomers of fructose, that is alpha D negative fructofuranose and beta D negative fructofuranose. Okay, so by mistake here it is written as apimers. Okay, they are actually anomers. So keep in mind, in ketosis, if they differ at C2, they are anomers. In aldosis, if they differ at C1, they are called five-membered ring structure of glucose is known as by mistake it is written five-membered ring structure of a alpha D glucose and beta D glucose are now sucrose. So cane sugar is sucrose, and there is a glycosidic bond between glucose and fructose, and that's why as there are two monosaccharides joined together by glycosidic linkage, it is called disaccharide. And you know both the reducing groups, C1 of glucose and C2 of fructose, they are bonded together, and that's why they are called non-reducing. Glucose gives on hydrolysis glucose and fructose. This is alpha D plus glucose, this is beta D negative fructose, sorry, fructofuranose, beta D negative fructofuranose, and C H and OH. And together, it is written upside down. Okay, this one. That's why all the groups are interchanged. See, this H and OH is removed, water is removed, and we get glycosidic linkage. Why it is called invert sugar? Because glucose is dextrorotatory, but after hydrolysis it gives dextrorotatory glucose and levorotatory fructose. And see, the levo-rotation of fructose is minus 92.4 degree, but dextrorotation of glucose is plus 52.4. If we see the yes, what we do, if it is in equal amount, specific rotation is minus 92.4 plus 52.5 divided by 2. That is the specific rotation of the mixture. As a result, sucrose brings about a change in the sign of rotation from dextro to levo, and that's why product is known as invert sugar. Now, maltose, that is obtained by the partial hydrolysis of starch. Bond is formed between C1 1 and 4 carbon of a glucose molecule. Is C1 of one glucose unit and C4 of the another glucose unit. That's why it is a reducing sugar, because one of the C1 is free. They are one alpha D plus glucose, beta alpha D plus glucose. Both are alpha D plus glucose. Copyranose both bond together, and this C1 is free. As this C1 is free, that's why it is reducing sugar. At least one C1 should be free in glucose or C2 in ketone. Lactose. So it is obtained from beta D plus galactose and beta D negative glucose. Glycosidic reaction between C1 of galactose and C4 of glucose. And this is also called milk sugar. Again, it is a disaccharide and it is again reducing sugar because C1 is free. See, it is epimer. See, in the glucose, see C1HOH, HOH, OH, H, HOH is glucose, but here OHH. OHH is, so in beta OH should be on the top, right? OHH, HOH, see this one C4. There is a difference in C4 configuration. Here it is HOH, here it is OHH. So galactose is C4 epimer of glucose. Same. Lactose is C4 epimer of glucose. Okay. Got it? And this is called milk sugar, again a reducing sugar. In what sugar is a mixture of yes, option E, glucose, and L, fructose. This small d and small l are dextro and levo. This small d and small l, it means dextro and levo. Capital D and capital L, that's different. That is configuration. This is rotation. Glycosidic linkage between question is incomplete. I think glycosidic linkage in sucrose. It is not sucrose. It both can be option maltose or lactose. Structure of polysaccharides and importance of carbohydrates, that is the last part of topic. Structure of starch. Starch is a polymer of glucose, alpha D glucose, and cellulose is a polymer of beta D. Starch consists of two fractions. One is amylose and other is amylopectin. These are the two fractions of starch. Consists of two polysaccharides, we can say. One is amylose, other is amylopectin. Amylose is only 20%, which is water soluble component, and amylopectin is 80%. Amylose contains 200 to 1000 alpha D glucose units, and there is C1 C4 glycosidic linkage, just like maltose. Means it is extension of maltose, we can say, linear chain of alpha D. Write those units side by side and just remove water. This way there is a glycosidic linkage, and so on. Is this extension of a maltose. They are called alpha glycosidic linkage. Amylopectin, first we have amylose unit, alpha linkages, another amylose unit. When these two amylose units, these amylose units, when we have C1 of first chain and C6 of the other chain, C1 of first chain and carbon of first chain, okay, and C6 of the other chain, the branching in two amylose molecules. Write one amylose chain and second amylose chain, and then attach C1 of the first chain and carbon of the first chain, and C6 of the any carbon in between the chain, and we get this branched isomer. And see, amylose is water soluble, amylopectin is water insoluble. Major component of plant structure is cellulose. There is long unbranched polymer. This straight chain. Cellulose consists of two glucose molecules joined by, now there is beta linkage, okay, beta glycosidic linkage. In starch there is alpha glycosidic linkage. So cellulose consists of linear chain of beta D glucose units. It is straight chain, but actually in the book it is written like that only, but it is again a straight chain like this. It is in zigzag fashion. This is beta linkage. Now glycogen, that is also called animal starch. Carbohydrates are stored in animal body as glycogen. Sources are liver, muscles, brain, yeast. Structurally it is similar to amylopectin, but it's more complex, more branched than the amylopectin of starch. That's why it is stored in the body. When we need glucose, times breaks the glycogen down to carbohydrates. You know, it is very essential for life. Both plants and animals. Honey is used as an instant source of energy by Veds in Ayurvedic. Carbohydrates are also used as storage molecules. They are used Hello. Hello. Wait, wait. Okay, carbohydrates formed in biosystem combination with many proteins and lipids. Or the link between glucose units and starch. Alpha glycosidic. There. Amylopectin is a polymer of amylopectin. Yes, both amylose as well as amylopectin. They contain alpha. Yes, but amylose in at the end there is what is monosaccharide in amylose as well as amylopectin. Okay. Okay. So the questions, previous year questions. What are the correct structural descriptions for M and N? Is alpha D plus glucopyranose. Okay, because we look at this OH is below the plane. Beta D negative fructose. That means it should be option. This is from ICER or a nest. I don't know, but I have collected. I don't know, but I have collected questions from previous years. Okay. Question next is structure of a disaccharide M. Its molecular mass is 342. Obviously, it is either maltose or lactose. Sorry, lactose. So which one is of them is this one? Yes. Yes. That means it is sucrose. So that means it is a combination of C1 of glucose and C2 of fructose. So M is a non-reducing sugar or reducing sugar. That means A option is correct. On hydrolysis, M gives one aldohexose and one aldopentose. True? Yes, one aldohexose and other is ketohexose. That means Second option is incorrect. If the specific rotation of x, y and m are given, for x it is given as plus 52. So obviously this is x, should be glucose, and for y it is minus 92.5, but it is hypothetically given as minus 74. Okay, that means mixture of x and y upon hydrolysis is laboratory. Is it true? For m it is plus 29. Of mixture of x and y, because it is, I told you, plus 52 minus 74 divided by 2. It is negative. Is true. Now 0.171 gram of m requires 0.153 gram of acetic anhydride for complete acetylation. This is stoichiometric questions. See, acetylation using acetic anhydride is one of the method used in structure determination of a carbohydrate. Yes, yes, we will count the number of OH. 1, 2, 3, 4, 5, 6, 7, 8. But at the same time, you know, this glycosidic linkage also breaks when there is acetylation. When it breaks, it also gives 2 OH. Got it? That means there are total 10 acetic anhydrides, right? That means 1 mole of M is equivalent to 10 moles of acetic anhydride, right? So 1 mole of M, its mass is given, and 342 gram of M, it is equivalent to 10 into, what is the molecular mass of this one? (102), right? That means it is equal to 1020 gram. 342 gram of M is equivalent to 1020 gram of acetic anhydride. What if it is 0.171? upon 342 to 0.171 comes out to be so 342 grams gives 1000 requires 1020 gram. 0.171 gram of M requires 0.51. 0.0. That means it is not true. Oh, yes, yes, yes. That means A and C options are correct. This question is from is asked in the exam, but it is from the COVID side. You know, in DRDO collaboration with Dr. Reddy's lab, it give this anti-COVID-19 drug 2-DG, 2-deoxy D-glucose. Same, I think, ICER or NEST. Polysaccharide that is used as a storage molecule in animals is glycogen. In aqueous solution, glucose exists in cyclic and open chain forms in equilibrium. As it is shown, glucose solution does not give positive Schiff's test, that you know. So, what is the correct statement out of these? Alpha D plus glucose and beta D plus glucose are enantiomers. Alpha D plus glucose and beta D plus glucose are anomers. In option B is correct. Yes, because say in solution open chain structure predominates over cyclic. Wrong. They are not enantiomers, right? Glucose reacts with sodium bisulfide. Wrong. Does not react. But it does not react with it as NH2OH. Glucose on treatment with bromine water yields X. What we get? COH on the first carbon, right? And X upon treatment with excess of acetic anhydride will give H, OH, OH, H, H, OH, H, OH and CH2OH. Here all cases will write this OAC should be the, in the first case, in second case it is okay, but here it is wrong. Here this case it is like this, right? Then it should be like this. Okay, so that's all for the day. Thank you. Next time we will do amino acids and proteins. Okay, worries, we'll cover it. Yes, yes.